I was actually going to present on a different topic. I was going to talk about respiratory function grading scheme because over the last year I have been working on a large project where we analyzed thousands of results from the RFG scheme. But I really wanted to have the paper published first, and as luck would have it, the paper was formally accepted last week. So I would very much ask you to keep an eye on +1 because over the next few weeks you should be seeing a paper that I believe will be very, very interesting to all of us. But I will be talking today about genetic diversity. We've already had a really, really great talk from Paula and Ariana, and I completely share their enthusiasm. So just to give you a little bit of background on myself, I joined the Royal Kennel Club 5 years ago, but before that I worked actually at the Roslin Institute, and my background is very much in genomics. So I really deeply hope that we will move to genomics. But for now, I'll talk about genetic diversity from the point of view of a kennel club, as we are. First, though, I would like to take a step back and talk a little bit about diseases in dogs. So we've heard a lot about conformation-related disease and extreme conformation in dogs over the last couple of days. However, we do need to remember that actually they form minority. Majority of the conditions that we see in dogs are unrelated to breed standards and conformation, and most of the known conditions at least, are simple monogenic conditions. And over time, we have seen some very encouraging results. Ross, just a moment ago, showed us the decline in the frequency of mutations. We had a previous paper published in 2018, which showed the same patterns across multiple mutations and multiple breeds. We have also seen an improvement in the complex diseases such as hip dysplasia. So here we have a comparison of Newfoundlands in the first row, born in 1990s early, and then 2010, and in the second row we have Labrador retrievers. So what you are seeing here is the distribution of the hip scores according to the UK system. So the 0 hip score is the best. The worst one is 106. So as we compare the current, well, a few years maybe out of date, but very recent population, we see that we have reduced the number of dogs with the high scores and increased the number of dogs with the low scores. So clearly it is incredibly rewarding to see breeders use tools such as DNA testing or hip scoring or estimated breeding values to reduce the prevalence of disease. However, very frustratingly, as we reduce the frequency of one disease, another one is identified. And that's not an issue. It's not related to any specific issues in dogs. In fact, the mutation rates that we see in dogs are typical, very similar to what we would see in livestock. And the study by Jonas Donner from a few years ago, I think it produced a really enlightening picture of what is actually happening. So by looking at over 100,000 dogs, they found that the mutations that we found in purebred dogs are also present in mixed breeds. So from the point of view genetics, it's totally understandable. But the reason that we're seeing a higher prevalence of disease in purebred dogs is that they are more likely to be homozygous for the deleterious variant, so have 2 copies of a recessive variant, which then results in the occurrence of a disease. This, of course, is a direct consequence of limited genetic diversity within pedigree dogs. Now, we've already heard about that, so I'll just very quickly talk about the factors which affect the genetic diversity. Well, pedigree dogs are typically close populations. I would say that they are not as close as most people believe. There are certain exceptions, and I think that we all know that there's also some illegitimate outcrossing happening. But as a rule, yes, we can describe them as close populations, which means that the genetic diversity that we are dealing with is a subset of the variation that was present in breed founders, and those founders typically were very few. And then over time, unfortunately, that diversity has been further reduced by population bottlenecks, chance events, selection both for health, conformation, and maybe working capabilities or temperament. And of course, harmful breeding practices such as use of popular sires that Ross just talked about a moment ago. So all these factors leads to reduced genetic diversity, and as we've said, that contributes to the expression of recessive diseases. But importantly, it also reduces our resources for selection against known problems, especially those that require collaboration, let's say, of many, many different genes. So actually, a proactive active approach to sustainable breeding should include both a selection for health and management of genetic diversity. And in practice, unfortunately, I would say that we tend to choose the reactive approach. So whenever a new condition is identified, we focus on that condition, we use intense selection, and we don't really pay attention to what is happening to genetic diversity while we're doing this. And this approach may give us short-term gains, and that can be very rewarding initially, but it is a very risky strategy for long-term sustainability. And now, of course, dogs are not the first species to face challenges related to genetic diversity. And you've already heard about livestock. I also included the conservation species here, where the situation is quite a bit different, because in contrast to livestock, where we have selection for multiple different traits, including productivity, but also health, fertility, the reproduction in general. In conservation species, there is no selection, other than natural selection, of course. All the focus is on maintaining variation, and as far as I know, there is no outcrossing, or at least no human-directed outcrossing. As compared to livestock, where outcrossing is used as a tool when appropriate. But what these two groups of species have in common is that typically there is few stakeholders with large numbers of animals. And as Ariana said, the breeding programs are very, very centralized. So with dogs, the genetic principles are exactly the same as in other species. We have genetic diversity, we lose it through certain processes, and there are very few ways in which we can reintroduce genetic diversity to a population. But the problem is that our population structure is completely different. And that means that we need new solutions. So those solutions have to be based on a firm understanding of genetics, of course, but they do need to account for the very unique structures we have in pedigree dogs overall, but also within breeds. And a very important point that we need to remember about from the beginning when we're talking about management of populations is that it's really not how we manage genetic diversity, it's more how we convince breeders to move from the litter focus to collaboration and collective responsibility for entire population. So when I took on this project, I decided to start at the very beginning, which meant undertaking a thorough analysis of the populations we aim to manage. And my initial plan was to basically update analyses that were carried out by my predecessor, Tom Lewis. He published a paper in 2013 where he described the analysis, and at that time he looked at average inbreeding coefficient and its changes over time. He estimated the effective population size, and he checked for the evidence of popular sires. So I was going to do the same thing, but as is usual with me and data, when I started playing with the data, I just started adding extra bits here and there. So when it came to genetic parameters, I also looked at the average relationships and the cohorts of dogs that we have currently in the country. I did look at the effect of inbreeding on litter sizes. With sires, I looked at their characteristics. So I was interested in seeing whether among the routinely collected data, is there anything that would help us identify those sires that are likely to become popular sires in the future? And then I also looked at the whole range of demographic parameters because even though not strictly related to genetics, they will have a huge impact on any management strategy. So I carried out the population analysis individually for 222 breeds that we recognized at the time. I used the entire electronic pedigrees to calculate the genetic parameters, but the demographic data was calculated for the UK registered dogs only. And the results were placed in breed reports. So this is an example, I believe it was a Gordon Setter. And these reports were shared with the breed communities. Now, I'd like to make it very clear that these breed reports were basically summaries of results. We did not provide them anything like, this is the action plan that you should follow. And this is partly because following the sharing of the breed reports, we had workshops with breed communities which were incredibly useful. And even though we expected that there was far more complexity than we could capture with our data, those meetings really revealed just how varied the breed circumstances can be. Nevertheless, as a kennel club that is supposed to support large numbers of breeds, we were wondering whether there were some common trends that we could use to guide our broader strategies. So in parallel to the within breed work, I also ran a cross breed analysis where I treated the breed estimate. So each breed was treated as a sample of a single pedigree population. So I used an approach called meta-analysis. And basically, we were trying to see whether there are any trends that are true for this hypothetical single population, and if there is any variability between breeds, how significant it is. Thanks to this approach, we identified a number of consistent trends across breeds. And we have published the demographic parameters to date. So the main demographic results were the pedigree dog population is declining in size. We have small percentages of dogs used in breeding. We have a decline in the use of dogs successful in conformation shows in breeding. And we have an increasing reliance on imported dogs. So if you'd like to read about those results a bit more, I would invite you to find our paper. Also, I did a webinar, which you can find on YouTube in our health webinar series. What we haven't published yet is the meta-analysis results looking at the genetic parameters, but I mean, I'm going to share some snippets with you here. So the first one was we confirmed that the inbreeding rates are declining. So this is the plot that we see at the top here. The subplot A shows the inbreeding rates by period, and this was calculated for all breeds that had at least 50 dogs born per year. And the subplot B shows the same rate of inbreeding in top 10 breeds only. So we see that over time, we definitely see a decline in inbreeding rates, which is positive, which is likely caused by breeders opting for matings resulting in lower inbreeding and also the use of imported dogs in breeding. However, we also found evidence of inbreeding depression. So this is shown in the bottom box plot here. So on the horizontal axis, we have the coefficient of inbreeding, increasing values. And on the vertical, we have the litter size. So you can see that although there is a considerable variability, the higher the COI, the lower the litter size, which is really what we would expect. But very importantly, we also found that our current populations tend to have high average relationships. And this is very important because that means that avoiding inbreeding in the future is going to become more challenging. So the population analysis provided us with a whole huge bunch of results. But what is important for this particular conversation is that, first of all, we do have some trends that we can use to discuss the general kind of state of pedigree dogs. But there are significant differences that we need to account for when we are planning our management strategies. We know that breeders are minimizing the inbreeding in their litters, which is great. It shows the increasing understanding of the risks associated with inbreeding and limited genetic diversity. But we also have an indication that this will become more difficult in the future. Apologies for my dog barking in the background. We also have found that there are new criteria that are deciding on the attractiveness of dogs. So in the past, pretty much all popular sires were champions of either conformation shows or maybe working trials. Now that percentage of popular sires that are champions has dropped significantly, and there are new criteria that people are using, such as coat color or even the health test results of a particular sire. And we will talk about the increased use of imports in breeding for two reasons, really. The first one is, again, it shows the increasing understanding of the importance of genetic diversity. It shows that breeders are trying to seek novel genetic contributions to their breeding programs. But it also means that our pedigree-based estimates of inbreeding, of effective population sizes, of relationships, all genetic parameters are at risk of being biased if we are looking at shallow pedigrees that are often associated with imported dogs. We are very likely to omit the deeper relationships in those pedigrees. But also this means that with the increased transfer of dogs across the borders, we really need to start thinking about global population management strategies. So one of the key trends that I've just mentioned was that breeders are effectively minimizing coefficient of inbreeding. However, this also brings one of the major limitations of this approach. So it is very important that we all recognize what coefficients of inbreeding actually measure. So they measure the risks of a litter. So they measure the relationship between the parents of a puppy, and they hopefully minimize the risk of the puppy suffering from an inherited disease. And that is true for both estimates based on the pedigree or genetic markers. But what this doesn't do is it does not safeguard genetic diversity of the breed. And in fact, sometimes focusing too much on the COI of individual litters can result in a further reduction of the diversity. And this is very nicely illustrated when we think about popular sires that are imported. So let's say that this dog is genuinely unrelated to the population that we have in a country, and all the breeders are choosing this dog because they want to have a zero COI in their litters. And in first generation, they do achieve that because the sire is unrelated, whether we look at pedigree or DNA. So in that first generation, that is looking great because we have low inbreeding, but we also have low diversity because this dog was a popular sire. And when we start breeding this first generation into a second generation, that's when we get into really deep trouble because pretty much all the individuals in the first generation are half siblings. So our inbreeding immediately jumps to a minimum of 12.5%. So we end up with high inbreeding and low diversity. So we do need to remember that COI does not equal population management. The second limitation I've already mentioned relates to the shallow pedigree depth. And this will have a huge impact on our management strategies. And we've heard that genomics could be really, really useful here. And I absolutely hope that we will get there. And in fact, we are talking with some collaborators on methods in which genomics could be helping us in very critical situations. However, we do need to acknowledge that at this moment, population-wide data, genomic data, is not yet available, and majority of the genomic data is owned by commercial companies. And that is completely understandable because actually developing the technology is costing a lot of money. But what it means for people like myself who are trying to support breeds is that it is difficult to integrate the commercial data sets into coordinated population-wide programs. So we could get depressed. Or we could start thinking how we can change these limitations to from constraints to challenges that we're going to address. So having identified where the difficulties lie, we can now turn to how we begin to deal with them. How do we manage our populations? So the work that we will be carrying out this year builds on the population analysis, both within breeds and across breeds, as well as the workshops that we had with the breed representatives that I've mentioned. And in the talk about a couple of hours ago, you've heard from Alison about the feedback that she collected from the external experts. So all these channels were brought together in the Breeding for Health framework, the genetic diversity pillar. And as Alison has already said, the focus for us this year is the support for breeds with small population sizes. Now, in next years, we will be focusing on harmful breeding practices, and this will be primarily in the larger breeds. So breeds where you would think that given tens of thousands of puppies being born, they really should be okay with the genetic diversity. But unfortunately, the harmful breeding practices are affecting that diversity. And finally, we will have a special project on limited pedigree data because that will be having an impact on all breeds, large and small, and whether they appear to be at risk of suffering from the limited genetic diversity or not. So the overarching goal for us is for the breed communities that will be taking part in the small populations project to produce, own and follow their own breed management strategy, which will be based on both firm genetic principles, but also unique circumstances of each breed. And now I can see that Amy has appeared on my screen, so that means that I need to speed up. So I'm not going to tell you all the details on how we identify the small breeds, but if you'd like to know more about that, please do. I will just tell you that following a flow chart, so to speak, looking at the effective population sizes, the number of contributing dogs, and the number of dogs born in the breed, as well as listening to the feedback from the breed communities, we ended up with 100 breeds that will be taking part in this project. So for this year, we've planned 4 online meetings. The first one is on what is genetic diversity and why it matters. Second will be on breeding tools. Third on outcrossing. Fourth on the practical solutions. So I presented the plan, this plan of 4 meetings to the breed communities just to make sure that it covers everything that they would like to discuss. And I'm not going to show you all the results of the survey. However, I think that it's very important for all of us to know that in the order of importance, when we asked what are the ways in which we as the Kennel Club could be supporting the breed communities, the first one that breeders selected was guidance and advice. The second one was practical tools, and the third one was education for wider breed community. So this is exactly what we are planning on doing. We are going to support them with the expertise that we have within the health team. We are going to support them with education of the wider communities. We will be facilitating collaborations And we will also provide support in tool use, and where possible, we will be also investigating new tool development. And I did say before that the goal is for the breed communities to write their own programs. And this is because at the end of the day, they will have to take responsibility for their own population. And we believe that if we start from a place where they have the feeling of ownership, that will bring us a better chance of success. I'm really rushing here, but one final thing that I would like to mention is the need for international collaboration. So I've already said that before. For some breeds, international collaboration is going to be absolutely necessary. Also, I think that genetic principles are easy, but how we turn them into effective management strategies is difficult. And so we need a platform for brainstorming and exchanging ideas and educating ourselves. And because of that, I formed a international working group, which includes 28 members currently from 11 countries, as well as a FCI scientific committee and breeding committee. We have planned 3 meetings. I'm hoping that we'll continue beyond that, but we'll see what the group thinks. So this group is primarily consisting of geneticists and Kennel Club staff that is in the role adjacent to genetics. If you have not heard about this group and you would like to be involved, please do send me an email, and hopefully we will see you at the next meeting. I would like to say only that there is a little bit of an overlap, because what we have discussed during our first meeting, which was on the pedigree data integrity and data sharing, and that will be also discussed at the in-person meeting during the third theme. I think that Katarina is going to be leading on this one, if I'm not mistaken. So the discussion will be on the Global Canine Data Alliance, which is obviously a much bigger project. So to bring this all together, this is my last slide. We are all having shared challenges across different breeds, across different countries. We have small populations with limited genetic diversity. We have global movement and limited diversity and limited tools. But we can deal with that. We have resources that we can use as long as we collaborate with each other and freely exchange information and knowledge and breeding material. So with this, I will finish. I'm not going to read all the bullet points because I don't want to get Amy too angry. So thank you very much. And I appreciate everyone staying so late with this talk.